Welcome to everybody. Thanks for your presence. Today it's a big honor for us to introduce Professor David Stec from the Department of Physiology and Biophysics of the University of Mississippi. So many of us know Professor Stec thanks to his work in the field of mild hyperbilirubinemia protection against obesity and cardiovascular disease. But the areas of expertise of Professor Stec are larger, and in particular is involved in renal and cardiovascular physiology. And so the title of his lecture today is Cardiovascular Disease in Non-Alcoholic Fatty Liver Disease, Unraveling Mechanisms. So I thank again Professor Stec and leaving him the floor for his lecture.
All right, thank you so much for the introduction. Let me see if I can share my screen here. Okay. Again, thanks so much for the invitation to speak to this group. We all have a common interest in liver disease. And today I'd like to talk a little bit about a newer direction in my laboratory where we are really interested in the link between non-alcoholic fatty liver disease and cardiovascular disease. And we think we might have an interesting model in order to come up with some new hypotheses and test some mechanisms. First of all, non-alcoholic fatty liver disease, I don't think I have to tell this group, or what's more, the newer nomenclature is now metabolic associated fatty liver disease. Again, it's the most common liver disease affecting 20 to 30% of Americans and is clinically silent. And it is also related to obesity, diabetes, and dyslipidemia. Again, what you're going to see in my seminar is that this non-alcoholic or metabolic associated fatty liver disease increases the risk of mortality, with most cases of death actually due to cardiovascular disease, malignancy, or complications of cirrhosis. Last thing, to the extent of changes in livers associated with the increased risk of cardiovascular disease and increases in mortality. And then, again, we all know that this can transform into steatohepatitis, which can result in cirrhosis and liver cancers as well. So this is a summary of a meta-analysis of the global prevalence of coronary artery disease in non-alcoholic fatty or metabolic associated fatty liver disease. And you can see that there is a high degree of cardiovascular disease that is associated with the development of non-alcoholic fatty liver disease in several populations. This slide shows it's a German study which shows that non-alcoholic fatty liver disease was associated with the time to coronary heart disease in primary care patients. Again, the increased accelerated incidence of coronary heart disease is apparent in these patients that were diagnosed with non-alcoholic fatty liver disease. Not only is it non-alcoholic fatty liver disease can be associated with obesity, which often is in diabetes, but there's also a population of individuals that are non-obese, which also exhibit fatty liver disease. And in these patients, you can see that these patients exhibit not as much of an increased risk as it's seen in obese patients, but they do have an increased risk of, again, hypertension, diabetes, and dyslipidemia, sort of suggesting that somehow this non-alcoholic fatty liver disease is driving this incidence of cardiovascular disease, even in these non-obese patients. Not only can you look at cardiovascular disease in general, but when you look at heart disease specifically, diastolic function, there's a recent study that came out just last year, which documented left ventricular diastolic dysfunction. And these are young adults, less than 40 years old, with and without non-alcoholic fatty liver disease. And again, these are relatively young otherwise healthy individuals, and you can see that these patients exhibit alterations in diastolic function. Again, this E to A ratio, which is a indice of the filling of the heart in diastole, is not only altered in patients with steatosis and diabetes, but patients that only have steatosis, hepatic steatosis as well, exhibit alterations in diastolic function. So there is, again, suggesting there's a link between this hepatic lipid accumulation and the diastolic function of the heart. Lastly, not only is non-alcoholic fatty liver disease associated with cardiac abnormalities, but this is a study from 2017 that shows the increased risk of chronic kidney disease, or CKD, in patients with type 1 diabetes and non-alcoholic fatty liver disease. You can see that the probability of developing chronic kidney disease and having severe complications of chronic kidney disease is markedly increased in patients that exhibit non-alcoholic fatty liver disease. So it is clear to me that there is a real significant link between the development of non-alcoholic fatty liver disease and its effect on the heart and on the kidney. So how do we specifically test the role of hepatic lipid accumulation in the development of cardiovascular disease? So what would be a really good model that would be needed to study this phenomenon? Well, first of all, I believe that the model needs to be independent of dietary induced obesity and insulin resistance. So a lot of these models of, we have animal models of non-alcoholic or metabolic associated fatty liver disease, obviously are developed in animals with some type of dietary induced obesity, which to me can confound the effects of simple what is going on in the liver. What is the contribution of the hepatic steatosis to these cardiovascular phenotypes? I think it's important that the underlying pathology originate in the liver. Again, the fat accumulation that we see in these models should primarily be derived through alterations in hepatic lipid metabolism, such that the livers are starting to exhibit signs of steatosis. And most importantly, I believe in these models, they need to exhibit a similar cardiovascular phenotype as we see in NAFLD patients. Okay, so they need to have similar degrees of cardiovascular phenotypes in order to be a good model to translate. So our lab has a couple of different models, which I'd like to explain in brief, which I think could be good candidates for studying the role of non-alcoholic fatty liver disease in the development of cardiovascular and renal disease as well. We have two models in the lab. They're both derived from hepatocyte-specific knockout. One of them is of the gene PparaAlpha, peroxisome proliferated activated response alpha. Another one is the biliverdin reductase, or BVRA, that knockout mouse. We have already published what the effects of these mice, these knockouts are on the development of hepatic steatosis, but I'd just like to briefly highlight these. The first one I'll start with is a study we did in collaboration with my Dr. Terry Hines at the University of Kentucky. We looked at the role of specific deletion of PparaAlpha in the hepatocytes and what happened when we put these mice, again, on high-fat diet. What we saw in these mice is that in response to high-fat diet, the PparaAlpha, liver-specific PparaAlpha knockout mice developed higher degrees of hepatic steatosis as measured by non-invasive echo MRI, shown here in panel A. When we looked at hepatic triglycerides, the hepatic triglycerides were also elevated in this model. Not only are these mice exhibit increases in hepatic steatosis, but they also exhibit alterations in fasting insulin, above what we see in the control mice. Again, greater levels of fasting blood glucose and more inflammation as indicated by increases in plasma C-peptide levels. So this is a good model of increases in hepatic steatosis, especially in response to a high-fat diet. The other model that we have in the laboratory, and again, these studies were done in collaboration with Terry Hines' lab, is that when you lose biliverdin reductase specifically from the liver, these mice also accumulate lipid to a high degree. Again, this is increases in hepatic total fat as measured by non-invasive echo MRI, increases in our knockout mouse compared to the control animals, increases in hepatic triglycerides, and again, we showed by oil red O staining, increases in hepatic steatosis. They also have alterations in fasting blood glucose and insulin. The important thing that we saw, and again, this is demonstrated in our model of PparaAlpha knockout mice, and this is what got us initially interested in this model as a potential model for looking at the development of cardiovascular disease in response to looking at the effect of hepatic steatosis in response to development of cardiovascular disease, is even on a normal fat diet. And this is a study we published back in 2019. We saw that the PparaAlpha knockout mice have increased levels of hepatic steatosis, even on a basal diet. I don't have the data with me right now to show you, but we still see a similar effect in our hepatic-specific BVRA knockout mice, in that they also accumulate lipids in the liver, even when they are placed on a normal fat or a standard laboratory diet. So given the fact that these mice accumulate a good amount of lipid in the liver, even in response to a normal fat diet, we thought that they might be a good model in order to look at the cardiovascular phenotype and look at the effect of hepatic steatosis on the development of any type of cardiovascular or renal pathology. So in order to get to that point, it's a little bit of a complicated, or a little bit of a story. We were initially interested in these mice, these PPAR-specific knockout mice, for a project that goes back a couple years in my laboratory. PparaAlpha inducers, such as fibrates, have been shown to be effective antihypertensive drugs in several rodent models of hypertension. Both salt-sensitive and salt-resistant or essential hypertensive models, when you treat with PparaAlpha agonists, such as fenofibrate, they have shown to be able to lower and prevent the development of hypertension. My laboratory did studies back in the early 2000s, which in mice, in a model of angiotensin II mediated hypertension, such that we showed that when you pre-treat animals with fenofibrate, the PparaAlpha antagonist, you're able to completely prevent the development of angiotensin II induced hypertension. So in this model, we give angiotensin II in osmotic mini pumps at a pretty high dose of about 1,000 nanograms per kilogram per minute, which gives us a nice reproducible hypertension of about 30 to 40 millimeters of mercury as compared to our control animals. When we put these animals on, when we treat these animals with fenofibrate for a week before we expose them to angiotensin II, you can see that it completely prevents the development of hypertension in this model. Now, initially, this was work, again, done back in the early 2000s. I was really interested in the role of cytochrome P450 metabolites, especially this metabolite here called 20-hydroxy-eicosanoic acid, or 20-HEAT. What we've shown, or what has been shown about 20-HEAT, now the role of 20-HEAT in hypertension, I really don't want, I can give it a whole talk about this. I've done a lot of work in this area earlier in my career, but it comes down to 20-HEAT in renal tubule cells is a very potent natriuretic compound. And what we found is that mice in particular make very little 20-HEAT in their kidneys under normal conditions. And when you treat them with fenofibrate, it really markedly upregulates CYP4A proteins, which are the proteins responsible for the generation of 20 heat. And it increases significantly 20 heat production in this model. So what we found is not only when you treat with fenofibrate, you see increases in 20 heat production in the kidney, but also in the liver. And we were very interested in the role of renal 20 heat production in the attenuation of hypertension. And we believed at the time that when you treat with fenofibrate, it lowers blood pressure or prevents the development of hypertension through this effect in the kidney to increase 20 heat production. And 20 heat, again, is a potent natriuretic compound. It prevents salt and water reabsorption in the thick ascending loop of Henle, acts as a diuretic basically, and lowers blood pressure. So we have this model now of PPAR alpha knockouts where we're able to do knockouts in various tissues. So what we wanted to initially ask was, what is the role of renal versus extrarenal PPAR alpha induction in the antihypertensive actions of fenofibrate? So we have this model again. So we have an experimental strategy to separate the renal versus non-renal effects of fenofibrate on the development of, on the regulation of blood pressure by upregulation of PPAR alpha with fibrate compounds. And the model that we chose to use, or that we're using, is a inducible kidney model, which it has a LC1 Cre, which is driven by this RT reverse transactivator PAX8. And the RT PAX8 is supposedly active in the kidney, and it gives you a kidney knockout. And then we also have our liver-specific PPAR alpha knockouts with driven by albumin Cre to get a liver-specific knockout. So what we noticed in the literature when we started to look at this model of this kidney-specific model using this LC1 Cre/RTTA PAX mice, when you look at the original report of this model, you can see that this is a when you induce the knockout with feeding with doxycycline, you can see you really get a robust knockout of a robust effect in the kidney. This again is a beta-galactosidase activated gene. It's activated in response to knockout by Cre recombinase, and you can see that when you stain blue, that the kidneys stain indicating that it's active in the kidney. However, there's also a certain population of liver cells that are affected in this model. This is due to this model. PAX8 is a transgene, and I think when it got inserted in the development of this model, it just inserted in an area of the genome where it was able, not only, it's normally kidney restricted, but this insertion of this transgene in this model allowed for some ectopic expression in the liver. So we were sort of concerned, and we were in the process of developing this kidney-specific model. This kidney-specific model is basically you're making a double transgenic animal on a flox background. So it takes a little bit of takes a little bit of breeding to generate this model. And so while we were generating this model, I said, you know, why don't we look at the effect of liver-specific knockout of PPAR alpha, because it's going to be knocked out to some extent in this model. And, you know, we'll show that, you know, knockout of the liver PPAR alpha really has no effect on blood pressure. And so when we go to use our kidney model, even though it would have some knockout in the liver, if we prove that total liver knockout of PPAR alpha has no effect, then we can just discount this ectopic expression of PPAR alpha. So what we did is we did a experimental protocol on about 30-week-old male mice. The mice were put on a diet. We put fenofibrate in their chow at a dose of about 90 milligrams per kilogram per day for 1 week. After that 1 week period, we implanted osmotic mini pumps that gave them angiotensin II at a concentration of 1 microgram per kilogram per minute. We let the mice go for 1 week. Then we put, the mice underwent surgery for direct blood pressure measurements by indwelling carotid catheters. We measured blood pressure for 3 days. At the end of the 3-day period, we euthanized the animals. We collected the plasma samples and tissue samples. So the first thing we wanted to show in our animals is that what is the effect of this oral fenofibrate treatment on one of the hallmarks of these fibrates, again, the peroxisome proliferating proliferators, and they do cause increases in liver weight. And so what we found is that in our wild type animals, we saw a nice increase in liver weight in response to oral fenofibrate treatment up for 2 weeks, where our knockout mice, not surprisingly, had no effect. Fenofibrate had no effect on the liver weight in these animals. So since we saw an effect in the liver, we then knew that this dose of fenofibrate should be active, and we then looked at the effect on blood pressure. So this is the effect of fenofibrate treatment on blood pressure in wild type, which are in circles, and knockout animals. Under baseline conditions, what we found is that the liver-specific PparaAlpha knockout mice exhibited increases in blood pressure as compared to the wild type animals of about 10 to 12 millimeters or 15 millimeters of mercury. When we gave both animals angiotensin II, we really didn't see any response in any difference in the blood pressure response to angiotensin II. They both got equally hypertensive. When we give fenofibrate in our wild type animals, similar to what we saw in a previous study in my lab, we saw almost a complete prevention of the development of hypertension in the wild type animals. And much to our surprise, at least initially, because again, I was under the impression initially that, when you give fenofibrate, it's more of a renal effect, a direct renal effect on increasing CYP enzymes, causing natriuresis and lowering blood pressure. However, when you knock out PparaAlpha from the liver of these animals, you can see that you totally block the antihypertensive actions of fenofibrate. So this suggests that there is some critical component in the liver of these animals that mediates the antihypertensive response to fenofibrate. Although I don't have them on this slide, we have some preliminary studies in what we really wanted to do in our kidney-specific knockout animals. And what you can see in the kidney-specific animals, similar to the liver-specific animals, is that when you treat those animals with fenofibrate, you get no effect to lower blood pressure in those animals as well. So there is, again, I was hoping it would be a little bit more clean. Those kidney animals do have loss of PparaAlpha in the liver, but we're trying to quantitate what the degree of knockout that they have in the liver in that model. How much do we influence liver PparaAlpha in that model? And we're not there yet, but I can tell that those animals have a similar phenotype to these animals. And so I was kind of surprised, that the liver is mediating this part of this response is totally mediated by the liver in these animals. So that led us to think about, what are some of the mechanisms? How does the liver mediate the antihypertensive effects of fenofibrate treatment? Again, I was really focused, or my past research in this area, we really were focused on the kidney. We thought it was a direct effect to increase CYP enzymes, to increase 20 heat production. We haven't looked at that yet. I mean, that could be part of it. We haven't really looked at that in our renal model yet, but it's clear that there's an effect on the liver. So one of the things that we looked at, and this again was in collaboration with Terry Hines, he's really had some good ideas in this area. Was one of the things that he was really interested in this model was beta-hydroxybutyrate or levels. And beta-hydroxybutyrate is a ketone body that's produced in the fasting state. Its blood levels increase with ketogenic diets, okay? And it's synthesized mainly from the liver via metabolisms of fatty acids. And the thing about beta-hydroxybutyrate is that it's a very important source of energy for tissues. especially the brain and the heart, in the absence of glucose. So what we did is we're able to collaborate with Vanderbilt University. Interestingly enough, my brother runs the NMR facility, and one of the things that they're able to do is they're able to use nuclear magnetic resonance spectroscopy, or NMR, to look at plasma, lipids, and metabolites. Okay, and so what we do is we collect some plasma from our animals. We only need a small amount, about 50 microliters of plasma, and then we can send it off to him, and as part of his facility, they can use this Bruker IVDr system to give us a complete lipid and metabolite profile in these animals. And interestingly, one of the metabolites that they can measure for us by NMR is beta-hydroxybutyrate levels. This is the data we looked at. What is the effect of oral fenofibrate treatment on beta-hydroxybutyrate in our mice? Again, in control animals, the liver-specific PparaAlpha animals have a decreased levels of beta-hydroxybutyrate in their plasma as compared to wild-type animals. Interestingly, when you treat with angiotensin II, it tends to lower the beta-hydroxybutyrate levels in the wild-type animals. It really has no effect on the in the knockouts. And more importantly, when you treat with fenofibrate treatment in the wild-type animals resulted in a marked increase in beta-hydroxybutyrate levels, whereas in the knockouts, it had no effect on the beta-hydroxybutyrate production. So it looks like fenofibrate treatment can increase levels of beta-hydroxybutyrate production in at least in wild-type animals, and this response is totally lost in the knockouts. Not only that, is interestingly enough, our other model, the biliverdin reductase knockout mice, the hepatic specific biliverdin reductase knockout mice, also exhibit a decrease in beta-hydroxybutyrate production under basal conditions. However, these animals are totally responsive to increases in beta-hydroxybutyrate in response to fenofibrate treatment. So while they exhibit lower levels in baseline, our hepatic-specific BVRA knockout mice are able to respond to increases, are able to increase beta-hydroxybutyrate production in response to a fenofibrate treatment. Again, I'm going to give this later on in my seminar. We really haven't characterized any of the cardiovascular phenotypes in this model, but we are in the process of doing that in my laboratory. So why is this important, or why may this be important? Again, this is a study that came out a few years ago from a group at the University of Toledo, where, again, my collaborator, Terry Hines, used to work, and he was very involved in this study as well. In what they showed, again, this is a model of salt-sensitive hypertension, the dull salt-sensitive rat. And what they showed is that in response to high salt feeding, that beta-hydroxybutyrate levels in the plasma are decreased. And when you supplement with this precursor of beta-hydroxybutyrate, this 1,3-butanediol, you can rescue this decrease in plasma beta-hydroxybutyrate levels in both the fasting and the fed state. And more importantly, what they saw, and again, this is a model of salt-sensitive hypertension. When you put the animals on high-salt diet, you see that they have significantly elevated blood pressures. However, when you pre-treat these animals with 1,3-butanediol, you can see that this totally attenuates the development of hypertension in this model. So what we set out to do in my laboratory is we did a pilot study where we looked at the effect of 1,3-butadiol supplementation in our liver-specific liver-specific PparaAlpha knockout mice, and wanted to look at the effect on blood pressure. This is a study, again, a pilot study that we did. And in this study, we measured blood pressure by the gold standard, or radiotelemetry. So this is direct measurements of blood pressure 24 hours a day using radiotelemetry, which is state-of-the-art gold standard for measurement of blood pressure in mice. Now, the previous study that I showed you, we did indwelling catheters, again, in conscious free-moving mice, which again, in my opinion, is, you'll see we got similar results. But again, a lot of people argue that, they are hooked up to, when you look at direct blood pressure measurements, they are hooked up to pressure transducers. But this is, again, free-moving mice that are implanted with radiotelemetry. It's gold standard. And what we saw, again, is that the liver-specific PparaAlpha knockout mice exhibited increases in blood pressure, okay? So when we treated these mice with 1,3-butadiol for only 1 week, we put them on this for 1 week, and in response to that, their blood pressure fell in the knockout animals. It got normalized to the levels that we saw in the wild-type animals. And when we put the wild-type animals on 1,3-butadiol, it really didn't have any effect on their blood pressure. So we have yet to measure how much the beta-hydroxybutyrate levels increased in this model, but this is really exciting preliminary data for us to show that 1,3-butadiol supplementation in this model can correct the hypertension or lower the blood pressure in this model. Next, we wanted to really look at a little bit of the cardiac function in these mice. And these studies have been done in my laboratory under the direction of a new postdoctoral fellow, Alfonso Badmus, in my laboratory. And what Alfonso was able to do is she takes our hepatocyte-specific knockout and control mice. We have a Vivo 3100 echocardiograph sonogram with transducers, which are able to give us both parasternal long axis and short axis views of the heart to look at more of the structure. And then we can do classic M mode echocardiography to look at more of the function in this model. And so Alfonso has been looking at the cardiac function in this model. This is just some of the general indices of cardiac function. We see a trend towards decreases in heart rate and ejection fraction in this model, indications of maybe systolic dysfunction, lowering of cardiac output in this knockout model. Again, these are 30-week-old animals on a normal fat diet. So they're relatively, they're not obese. What we've showed before is that in a normal fat diet, these animals have normal blood glucose and insulin sensitivity. So again, but they do have signs that they may have some cardiac systolic dysfunction. They definitely have signs of markers of diastolic dysfunction. When you look at the E prime, the early diastolic mitral annual velocity, it's lower in the knockout, suggesting alterations in filling. When you look at the E to E prime ratio, or the indices of early mitral and inflow velocity to the annular early diastolic velocity, it's altered in the knockout mice as compared to the wild type mice. They don't exhibit any effect in isovolumic relaxation time, but they do have an alteration in diastolic filling. When we look at the ventricular studies in these animals, a little bit surprisingly, at least by echo MRI, we saw that they have a lower left ventricular mass. They have increases in left ventricular diameters in both systole and diastole, indicating incomplete emptying of the heart. And they also exhibit structural alterations. They have alterations in left ventricular anterior and posterior wall thicknesses, suggesting a thinning of the heart, like possibly a thinning of the heart due to hypoxia or ischemic lowering of the heart. So they do have alterations in this ventricular structure. So this model, again, as I showed you before, in NAFLD patients, they did have alterations in E to A. We have a tendency for that, but it's not significant yet. But they do have this alteration in diastolic filling in this animals. Again, this is only at 30 weeks of age. We haven't looked at older animals yet, and it's possible that this cardiac phenotype could be worse as these animals get older. One of the other things that has come out of the literature is that female NAFLD patients exhibit greater incidence of cardiovascular events and mortality. So this is a study, again, a retrospective study that was published in 2020, showing that, again, when you look at individual studies, it shows that, again, that female NAFLD patients are at greater risk for cardiovascular events and mortality. Again, they exhibit greater cardiovascular events and mortality with aging, again, even compared to males. So there's definitely a sex difference here. We've looked at this a little bit, again, in animals that are only 30-week-old, female animals, and we see a trend towards increases in blood pressure in our liver-specific PparaAlpha knockout mice at 30 weeks of age. And we are currently evaluating their cardiac phenotype in this model. It's possible, again, this is a lot milder than we see at the males. And we know that in human females, it takes, they have to be older in order for the phenotypes to be more prevalent. So it's possible that we may need to look at older aged females in this model as well. But we are currently doing that. Just in summary, again, what we're interested in these models, we really need to further characterize the cardiovascular parameters in our other model, the biliverdin reductase knockout mice, including blood pressure, cardiac structure and function, and vascular stiffness. We're also working on this in the liver-specific PparaAlpha knockout mice. We're using pulse wave echocardiography to look at that. And we are seeing what looks like a stiffer vessels in the PparaAlpha knockout animals. We want to, again, define the role of alterations in this beta-hydroxybutyrate via treatment with 1,3-butanediol. We had some interesting preliminary data, shows that supplementation, increasing these levels does lower blood pressure in this model. We need to figure out how. The previous study that I showed you from the group at Toledo seemed to indicate that beta-hydroxybutyrate supplementation altered immune cell infiltration in the kidney and lowered blood pressure in that model of salt hypertension. We haven't really looked at immune infiltration in this model yet, as far as whether immune cells are infiltrating these kidneys. We do have some studies planned to look at that kind of phenotype in our model, but we really have just got started on this. There's other pathways too that could be active in this system. One of them is, again, the renin-angiotensin system. We haven't measured, we are in the process of measuring plasma renin and angiotensin 2 levels in this model, see if it's elevated or altered. And we can, again, check out whether angiotensin 2 receptor blockers could be beneficial. A paper came out last year in the literature which really showed that this enzyme aldehyde dehydrogenase, or ALDH1A1, is upregulated in the liver of in NAFLD. And we know that this can be associated with changes in blood pressure. So we're looking at the role in our model of ALDH1A1, see if it's altered. And there are good inhibitors of this enzyme that are available, and we could treat animals and look at the effect of that. And lastly, what we really haven't looked at, but I'm very interested in, is the hepatic CYP metabolites For example, 20 heat, also epoxy eicosanoic acids or EETs. There's a possibility that these metabolites derived from the liver could be acting on the kidney to affect renal function. And so we're trying to look at the profile of hepatic CYP metabolites in our model of liver-specific PparaAlpha knockout. So in summary, again, we have these alterations in hepatic steatosis, which lead to things in the liver, maybe alterations in the renin-angiotensin system. We know that beta-hydroxybutyrate levels change, CYP metabolites. Are these directly affecting blood pressure? Is it through the kidney, through some inflammatory pathway? Do these changes directly impact the heart? Again, what is the effect directly on the heart of these things? So we have a lot of work to do to characterize a lot of the effects of these treatments and these potential mechanisms. But I think I'd like to get feedback from the group, what everybody else thinks. I think this is, a good model to look at the effect of just hepatic steatosis independent of a lot of these changes of like obesity and insulin resistance on the effect on the development of cardiovascular and renal pathology, and see what happens when we try to, does B-OHB treatment lower the hepatic steatosis? Does it have any effect on that, or are these pathways independent of the degree of steatosis? So these are all things that we need really to look more closely at in our model. And just in closing, I'd just like to acknowledge the contributions of a lot of individuals to this project. Alfonso Badmus, again, is the postdoctoral fellow in my laboratory who's really spearheading. She did all of the echocardiography work. She did a lot of the work with the direct measurements of blood pressure with telemetry. She got good at putting these telemeters in and analyzing the data. Alex De Silva in our laboratory, in our department, is helping us a lot with the echocardiography studies. Our assay core measures a lot of different things for us. Again, I'd like to acknowledge Terry Hines, who's a longtime collaborator of mine. He's been really instrumental in some of these pathways that we're looking at, and his expertise in the nuclear receptor signaling is critical to development of these models. Lastly, my brother at Vanderbilt University, who does the measurements of plasma metabolites and lipids for us. And then we have some collaborators at the University of Mississippi, which I didn't get into some of their work, but they really help us a lot with coming up with things to chemicals to look at the effect of bilirubin, which I really didn't talk about, but is an active component of our lab, not only my lab, but collaboration with Terry Hines. And again, support from the NIH, NIDDK, and also the National Institute of General Medical Sciences. And I really appreciate your attention, and I'm willing to take any questions.
So thank you very much, Professor Stec, for your excellent and very detailed presentation. So now we are open to questions. Are there any comments, questions? Libor, please.
Hi, David, again. So thank you very much for your presentation. I appreciate it very, very much. So I have one comment and one question, if I may.
Sure.
So the comment is that NAFLD is, as we all know, really a big group of diseases. It's not just one condition. And it was clear also from your presentation. I would like to mention that today there is a paper published in Lancet Gastroenterology and Hepatology, co-authored by 1,000 people, including me and Claudio, which is related to the, let's say, a redefinition of NAFLD, which would be called metabolic dysfunction-associated liver disease. And I would also like to mention that we published a week ago in Hepatology a paper, a multi-center collaborative project on different subtypes of NAFLD patients in relationship to the cardiovascular diseases. And there are at least 3 metabolomic subtypes which differentiate among each other in terms of risk of cardiovascular and atherosclerotic diseases, which is completely in agreement with what you said. And my question is that I would be very interested in knowing whether you try to investigate the role of glutathione in the pathogenesis of NAFLD, and maybe with the interaction with bilirubin, which, as we know, plays an important role.
Yeah, thank you very much for the question and the comment there, Libor. No, we haven't really been thinking about that, but that is a good idea. One of the things that we're interested in, and I understand, again, that this disease is a combination of a lot of different factors. One of the things that we're sort of interested in, that I'm more interested in, is, again, it's clear that diabetes and obesity contributes to cardiovascular disease, and we know that these patients have that, and it's definitely a component. But what I'm sort of trying to get at with these models, and I think we can look at, is what is the contribution of just the hepatic steatosis? What is is it important or not? Because if it's not, then maybe we don't really need to worry about it in some of these patients. Maybe we can treat them to treat the cardiovascular disease without getting at the underlying root. But if it is important, if it does drive at least a certain amount of the cardiovascular phenotypes that we see in this, how can we ever tell that? Well, we need to get a model that we can separate out these things, at least an animal model, and hopefully this animal model will have a phenotype that's similar to what we see in patients, and say, okay, well, this animal model doesn't have these other complications. So what if we just focus on the steatosis in these patients? If we focus on the steatosis, independent of changes of any of these other parameters, would that be beneficial? And again, it might be a stretch in humans to do this kind of thing, but I really, really hoping to see what is the effect of the underlying steatosis, and is it important to treat that, or should we just focus on, the cardiovascular disease and not necessarily worry if they lose lipids in the liver? Fine. If they don't, that's all good as well. So, but no, we haven't really looked at glutathione, and that's a really good, a possibility of one of the systems we should look at in our model.
Well, I mean, again, congratulations. That was really very informative. A well done wrap up of the different ideas and the data, a lot of data. You should be congratulated for that. I have a question following up what Libor mentioned. I mean, I think that your models, I mean, the animal models you have, may be also used very nicely to understand if there is any role of bilirubin in protecting the damage. Because you have the damage, and then you can play around with the level of bilirubin. This is most my question. Is the level of bilirubin in these animals equal? Is the same? Is higher? Because there is one thing which actually, it's important to mention. You mentioned that in females, all females, the effect is higher as compared to young females, if I got it correctly. So,
right, and humans and males too.
Exactly. If you look carefully in female, the normal amount of bilirubin in normal females is half than in the normal males. And I think this maybe play a role, as suggested also by Libor in different review papers. So I think that this could be an excellent idea to, to see whether by modulating the bilirubin level by drugs or whatever it is, or by crossbreeding with the so-called Gilbert mouse model, so-called, which is not the right one, but something, you can really modulate the effect on the cardiovascular disease. Because as pointed out by Libor, steatosis and the MAFLD, rather than NAFLD, it's just a sort of a umbrella under which it goes almost everything. Hypertension, insulin resistance, obesity, and you name it. And I think that having the different roles of each components dissected will be very important. And on top of that, this bilirubin may do something, as we all know can do. That's going to be wonderful.
Yeah. So let me start by, first of all, in the BVRA, liver-specific BVRA knockout mice, they have hepatic steatosis, you know, accelerated hepatic steatosis, but they don't have any changes in circulating bilirubin levels that we can measure. We did do a study, and we're writing it up right now, where we looked at bilirubin treatment in the liver-specific PparaAlpha knockout animals. And when we treat those animals with bilirubin, obviously the control animals that we, wild-type animals that we treat, bilirubin lowers hepatic fat, lowers blood glucose, upregulates a lot of stuff. But in a liver-specific animals, bilirubin treatment has no effect. It doesn't do anything to the steatosis. So I would imagine in that model, if bilirubin treatment wouldn't be very effective in that model if it's having its effect on the liver per se. Now, bilirubin can be having effects on kidney or the adipocytes to impact the cardiovascular function in that model. So we really haven't done that, but we do have. Again, part of my R01 grant that I have is looking at the cardiovascular effects of bilirubin treatment in animals on a high-fat diet. And the problem with that is that the animals that are on a high-fat diet, at least the C57 animals in our hands, they don't get much hypertension or, you know, kidney disease. They get really fat, but they don't get much hypertension. So it's not a good phenotype. We're looking for models that are a better phenotype to check out bilirubin. For example, there is a model called the BPH2 mouse. It's a spontaneously hypertensive mouse. And you could put them on a high-fat diet and they will get obese. I mean, they don't get as obese as normal animals for whatever reasons, but that's a model that we can look at bilirubin treatment in. So the problem in normal animals, at least in our hands, and I think the literature shows, is that they don't get very hypertensive. These C57-based animals, when you put them on a high-fat diet, their blood pressure doesn't go up that much, even though they get greatly obese.
Okay, thank you.
Yeah.
Are there other questions? If not, I think that we can thank again Professor Stec, and hoping to see you very soon in another situation as this.
Yeah, thanks everybody for attending and for your attention, and I appreciate the opportunity to present to the group.
To you. Congratulations, very nice.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye. You take care.
Yeah.
Thanks to all of you.